can have a seat, then Sander can make a start.
Hello guys, I'm really glad to be here. It's fun that I'm also talking a little bit in my own office. As you may know, next to here we also do our sampling, and I will tell you all about that. Als ik nu links-rechts ga blijven zitten.
Maar heb je de... Volgens mij gaat het goed, toch?
Oh, ja.
Maar heb je, wil je de klikker?
Ja, dat kan ook. Maar ik kan het ook zo doen. Maar niet.
Ja, de klikker moet hier ergens liggen. Want Francesca misschien. Ja?
So, I will start with introducing myself. I started off as an aquarist, so I always tell people that I'm a zookeeper that got out of hand. So I started working at Burger Zoo, and over there's one of the biggest reefs that's not connected to the sea. Of course, it's artificial, but I think it's a good living lab to actually learn more about also the things that Francesca told us a little bit about. But of course, Francesca does it for nature conservation, and for us, it's for the public. But I also worked for the WWF Sharks and Rays back into the North Sea project, which gives a little crossbreeding in my head too of how you can use the work as an aquarist for nature conservation. And now I'm working in the Rotterdam Zoo, and as Jamie also said, we can't let go of that stuff. So just as Jamie, I have an aquarium in my living room. That was actually the first furniture that we moved there. So why focus on grazers? What do we know from reef aquaria? Well, if I have an aquarium at home, this is a picture for my aquarium. I also know that I need grazers to make sure that I can maintain the reef. And it's quite funny because here you see a small blackfish, and they also call them the lawnmower bladdy. And I already know that shows a bit of his function that he had on the reef. So they're a cornerstone on the reef aquaria, but they're also a cornerstone on the reef, but we just don't understand too much about what they actually need. So we only know the role of the fish themselves. So what we know from corals is, of course, it's a slow-growing animal with a plant living inside, if we say it very basically, and they will always grow slower than plants. So you need something to take control of this pressure. And for example, fishes, snails, sea urchins are really good at taking these pressures away. But as I said, it's very hard to measure the extent of what's needed from them. So what I'm trying to do is always think back and make sure that what we know from land is also transferred to the sea. So I always say we need to get out of the helicopter to learn more about the fish behavior, about spawning behavior, and we also need more knowledge about feeding behavior. Because actually most fish that we know have a role on the reef, and they're very important. Certain fish are the gardeners of the reef. But for example, the groupers are the rodent killers to make sure that the rodents that can do harm, they also do in cities, but then underwater it's exactly the same. I always reference to parrotfish. Since 2 years, we know that a parrotfish actually hunts for microbacteria, cyanobacteria, to make sure that it can digest algae. We only know this for 2 years. And that's fun because there's such a cornerstone species of the reef, and in the meantime, we don't know what its individual needs are. And especially now in reef conservation, I think it's very good that we can already plant our corals, and Francesca showed us an incredible work. But if we take the fish and the sea urchins and the snails for granted, probably we will have a problem in the meantime. And hopefully we can help these guys out too. It's a shame that the title says, can we find aquaculture Dory? And that's very hard because working in an aquarium, I can say that we don't understand fish too well. Dory is only bred since 2017. It's actually a Picasso surgeonfish. And I think that already shows how less we know about the sea. So the role of the aquarium in the past was actually more taking away fish than bringing them back. And I hope there's a change there, because working in an aquarium also means that you can learn stuff about these fish and their roles that actually can help us preserve the reefs. So I came up with an idea. Why don't we try to breed fish in open waters? Because what we know from aquaculture is basically transferring fish to us and not adapt ourselves to reefs. And that's how we don't know so much. So for example, 80% of all fish spawn pelagically, which means the only parental deed is the deed, I always say. And in the meantime, we don't even know which egg corresponds to which fish. So we actually don't know anything about the first life stages of these fish and how we can protect and preserve them, or maybe even can make more of them. And there's some benefits of making it floating in open waters, because it will reduce energy. You don't need to heat or cool if you do it correctly. We already do a lot of land, and I think occupying more land space, especially on small islands like Bonaire, sometimes can be a very bad decision. And one very important thing that I can see in conservation, and especially in aquaculture, that we need to do is we need to have more uniformity in our work. And basically also share failures. I have a good example. I tried to breed a fish that's actually quite funny for the Caribbean that I tried to breed. It was the lionfish. But nobody ever bred a lionfish in the world before. I knew that. And I was trying it and I got it to eat back in the Rotterdam Zoo. And I thought I was failing because it always stopped at 8 days. So basically, I stopped trying. And then I went to the University of Florida, where they bred thousands of fish and many species for the first time worldwide. And these guys said, we don't know anyone that actually got them to eat. So actually I had a small success, but I wasn't able to share it because I didn't know it was a success. And I think if we start there, at more, how do you say, sorry, I forgot the word, nieuwsgierig. Yeah, if it starts with being curious about nature instead of the results come afterwards. And that's what I want to show you with this project. And the last sentence is the most important. If we already know the sea works, why don't we try to adapt ourselves to the sea? And I think there's a very big problem for us there, because what we know from corals can be transplanted to fish straight away. Because we were on an expedition. Because when you want to make sure that you can breed fish in open waters, you also need eggs. And before I had this ludicrous idea of trying to breed fish in open waters, it was more of a practical sense. And then I found out I actually made a tool that we can do very great work with. Because as I said, 80% of all fish are pelagic spawners. And there's more than 350 fish species on Bonaire. Before we went to Bonaire, there were 10 decent pictures in scientific papers of fish eggs that were pelagic. If you corresponded to the 300, we basically know nothing. That was the starting point for expedition. And because we make everything ourselves, and I'm very proud of that, it also is a very cool thing, because everything that we make in the Rotterdam Zoo is transferred to here. And the first steps we did here in Bonaire were funded by the WWF. And I can't stress this enough, that it's also a risk getting into something like that, because it's not science straight away. It's just the zookeeper that got out of hand that wants to try something, and in the end it was a tool. So I'm very grateful for the help that I got from the zoo, from DCNA. They gave us an office space, and the WWF, and of course also Stinapa, but I will tell more about that. So what we try to do now is focus on two roads. So we have this breeding system, but let's start with the most important part. If I don't know what I fill it up with, I have nothing. So basically we're trying to catch a fish egg, take pictures. We will take the pictures also in the afternoon with all of us. So it's a good test, because I only did this with junior rangers with Stinapa. So little kids, and I hope you're just as curious as them. That will be funny. And and afterwards we take a DNA analysis, because actually, if you don't know what you're looking at, you need to verify what you're actually doing. In the end, we try to we hopefully make an artificial intelligence database where we can fill up these pictures and help us make the right choice for DNA analysis, because now it's like finding a needle in a haystack. We really don't know sometimes what we're looking at. So as I said, we were starting to catch fish eggs. And the fun part is, the first time we did this with a group full of friends. So it was 6 people in one house. The house was also an office in the first time, because we weren't straight away in the DCNA office. And everything you see is hand built. So basically what we try to do is we build a frame, like a pump charge frame, to make sure that if a boat runs over, it still goes down and it doesn't break your nets, because these nets are very expensive. And we just put them to a Stinapa buoy. Very glad that we got the permits for that. And then we can lay it overnight, because most fish are spawning in the evening or in the morning. And then you have both in one swoop. And because you do it at one place, you also have places where you know that you catch these eggs. So in the end, what we try to do is not only fill up a database of eggs, but also a database of when it's happening. We also know that the moon has a big influence, as Jamie told, for corals, but it's the same for fish. But if you don't know where you're looking for, it's very good to try to have a comprehensive approach and already know places, moon cycle, and hopefully in the end know where to go to. Then we will take a picture. And here it's also getting funny, because most of the time in biology, if we want to take a picture, we kill it. Or it's something that doesn't move too much. So the fun part of what we try to do is already make sure that we can film and make movies. Because in the end, I hope to integrate this microscope camera also in our system, so we can track live what is happening. And I was going to the University of Delft, the technical university, and I asked if they had the right camera to help me around. And first they didn't know where to start and couldn't help me at all. So I found a camera that was for computer chips. If you want to make sure that the chips are still hot and you can melt them in the right position, you have to have a good autofocus, and it really helps us in taking pictures, but also movies. But I will tell you later about movies, because that's the coolest part. Afterwards, we take this DNA analysis. And this is very important, because otherwise we don't know what we're looking at, as I said. So we basically use the reference of adult fish, try to cross-reference, and see if we can find out which egg corresponds to which fish. And then you get this cool stuff. For example, from the French grant, we already had pictures in color, but most pictures that we can find in scientific papers aren't even in color. So what we try to do now is make sure that we fill it up with as much data as we can, because basically now we have a tool to measure biodiversity at the start of the life, not at how many fish we have, how many reproductive fish we have. And I think that's an important thing to start with, because it also is a different story. If you look at the presentation of Francesca and Kelly, you also see all of these eggs. And I always say an egg is a sign of new life and does give hope. And if you do the same with fish, then we can also show that there's still a way to bounce back, because it is still reproducing. So maybe the world is going to shit, but not as bad as not as bad as we think it is, because still there is some capability of regrowing yourself, and I think that's very important. Another cool one is greater soapfish. If you look this up in literature, we know it's there, we know it's breeding, but it's never even documented that it breeds. So basically, by taking this picture, this is the first time that we know in October at Waning Gibbous, this fish is breeding. And I think that also shows how less we know. We only look at adults. We don't look at the start of their life, which is probably the most critical phase in their life, because now it's just a swimming egg. So what you're looking at is actually almost the same as a fish egg, but then it has this oil globules to make it float. And this strategy also makes sure, because it's so tiny, for your explanation, this is 400 microns, so I think it's half a millimeter, and you can see through it, which means good luck this afternoon. It's very hard to spot them. To give you an explanation, we did our first try at Burger Zoo, and actually, because I was talking way too long, my former colleague Tamara found them because she was looking so long in the fish tank that you can actually see them. So it's very tiny, very different material than what we know. And what you also see, and I think that's already fun, these are all below 24 hours old. So you can already see pigmentation kicking in. But there's also a lot of things that you can't see with your own eyes. So that's why we want this AI to help us make the right choice, to select the right eggs. What we try to do now is actually make sure that it already measures by itself, so we don't need to do that all manually. And that will help us a great deal, because now we need to do everything by hand. So I'm really glad we have interns.
What's your skill?
Sorry?
What's the scale of the red line? How big are the eggs?
300 or 400 microns. So it's really tiny. And the fun part of what we are doing, I think, is also showing that all of these fish that live on different areas on the reef use the same way for reproduction. So basically a peacock flounder does the same thing while living on the bottom as a tuna does. And I also talk to the fishermen a lot, and there you also get some great results, because they finally understand in a different way, not with trying to teach them, but trying to learn together with them how the reef works. So if they are saying they're not fishing on a reef, I'm always saying, sometimes we both don't know, because we haven't been at the depths where these fish are. And if you catch a fish that is still breeding on the reef, there's still a correspondence. It's still one big ecosystem, and it's fun to see that you actually can work together in a different way, more practical, more pragmatic than before, because you actually, if you're looking at it, there's only 2 organizations directly working with fish, except from Stenapa, but it's a different role, working on fish in Bonaire, and it's Piscabon, the local fisheries cooperation, and us. And I think that also shows where we added society. It's a different role that we try to play in terms of conservation. Do it together with all these local partners, and not go straight away for the goal, because the goal is very far away. It will take a long while before we can actually breed fish in open waters. Of course, one of my favorites we also catch, it's always nice. We already had 2 surgeonfish. Another fun fact about a surgeonfish is that in papers they already stated in the 1980s that it's and it's the latest document about fish eggs, they already stated that it's hard to differentiate the egg of the blue surgeonfish and the egg of the ocean surgeonfish. And as you can see, there are already some slight differences. For example, the oil globule has a different color. But here you also come back to the point where it's getting hard. And when I talk about uniformity in the work, it's very important that we try to use the same tools to actually take these pictures. Because as you know, if you take your mobile phone and we would all take the same picture, it will look different. So how can we actually make sure that the science works for all of us instead of just for the paper? And that is my job. My job is to make a tool that others can make science with. And that's why we are already with 3 now. So we have Tim being the manager of the Caribbean Hub, managing all the projects. I'm more at the backside trying to create these tools. We have researcher Jeroen Ham now that will make sure that we actually prove what we're doing and also collect the data, because it's very hard to do this. It's a lot of handwork still, and we try to automate things. But as Francesca probably knows, the start of all these things is very hard. You do everything by hand and you learn by trial and error. We also got a first documentation of the Barrett Hamlet, which I think is also funny. I don't know if it corresponds, because basically we only put 10 pictures to the list. So now we're up to 20, which is cool. It doubled in size, but still that's less than 10% of the pelagic spawners known in Bonaire. Where we can already see that there's more pigmentation in some eggs where the fish also have more pigmentation as an adult. I don't know if there's a relationship, probably not, because in biology there's always some up and out, but maybe we can try to figure something out here. Here, as I said, we also had the skipjack tuna. That was a good way for me to go back to the fishermen and say, hey guys, I think you're just as dependent on the reef, even if you're not being there all the time. We also had a spotted drum that also had a fun thing, because most of the time there's no pigmentation in the egg yolk, and here you can see there's also pigmentation in the egg yolk. I don't know about you, but for me, my inner child is always happy, because it's actually quite the same as when you're going through your local lake and pull something out. But now we're actually seeing things for the first time in the world, which I think is the funniest thing ever. Another thing that makes our work harder, of course, is with the coral, you can put a net over it because it's not running around. And with the fish, that gives us the problem. That's why it's so hard to build up this database, and why we hope that if others are interested on other islands, that we actually can make sure that we do this together and make this bigger and better. Because having an understanding of these eggs and fish is not only about ecology or biology, you can also make new legislations with it. Because what we do now is just put out no fishing zones, while we don't know if it could also be the case that if you work together, for example, with the fishermen, when you know that the fish are breeding, you also select like we do now with lobsters. But the hard part there is if you touch that base, then it's a different conversation with the fishermen. So basically, I always say my role and our role is to show what is possible and not to implement it, because otherwise you're having 2 hats on. Another cool thing that we already learned a little from the aquarium, but we needed the DNA proof, is garden eels lay gigantic eggs. And it's funny, because if you know how small they are in the ocean, and when you look at this fella, and then go back to the tuna egg, because this is 0.5 millimeter, and this is also 0.5 millimeter, I think there's a big chance that larvae of this size can eat a tuna larva at the first stages of their life. I'm not sure, but how fun would that be? And basically, it's also so big, and we don't know if it corresponds, that it also has a lot of oil globules. And there comes the artisan knowledge, as I sometimes call it, that we have from an aquarium. Because in aquariums, we sometimes can see by the egg what it is. So the first time we collected these gigantic eggs, and probably you will also see them, they're 10 or sometimes even 20 times the size of the other fish eggs. You can see the difference in shape and size, and also why there's so much different strategies for these fish to breed. And there are similarities, but basically, and I will tell you more about it in the afternoon, after this one, there's the phase where we call it the hatchling, and it's just a swimming egg yolk. And the cool thing about this is that we also don't know too much about this phase. So during our expedition, we are not only looking at the eggs, but also at this hatchling phase, because I think if we know more and understand more about what is happening there, then we can say way more about where they end up as a larvae, because then the differentiation starts. But these first 6 days are crucial for 80% of all fish. So bear that in mind, because it's almost weird that nobody looked at it like this. So in the end, as I said, there's one big goal, but probably I will reach it when I'm gray, but you can't see it because I'm already bald. That's really nice. And and that's how can we make sure that we can make more of it, or actually proceed? Because I think there lies the most important part. It's not only about breeding, but also measuring. So I always say a breeding system is also a measurement system. I think many things that we learned from Jamie is also, if you know in captivity how it works, you can make it better. Well, there's a reason 85% of all marine fish living in an aquarium has never been bred before. So we need to shift our focus and make sure that we understand how the sea works, instead of trying to adapt everything to ourselves on land. And we already had some fun results with that, because what we see here, and this was pointed out by my girlfriend, and she thought it was a garbage bag from the top. I said, go and get it out. And then I got it out, and it was a big strain of eggs and gelatinous rabbits. And basically in there were 2 species, after DNA analysis probably, and there was more blennies, but also another fish that a lot of divers come for, and that's frogfish. And the fun part is that we already got it to eat without actually feeding it. And that's probably where I think the role lays, because in aquaria we can breed only 50 planktonic species, and we discover a new species every week. So we already had thousands of different species. So what I always say is, we are trying to feed a kid peanut butter while it likes jelly. It doesn't work like that. And again, we should adapt ourselves to the sea instead of adapt the sea to us. So what are the next steps? We need to create uniformity in our work to get a comprehensive egg database. And I think we can start doing this all over the world, because it doesn't matter if you're in the Caribbean, in an aquarium, or in the Indo-Pacific, Red Sea, maybe even the North Sea, it doesn't matter. Most fish eggs float. To get a proper understanding, start catching fish eggs. Take a picture, take a DNA analysis. If you know this, you can make a spawning calendar and actually understand better how this fish works. You can make legislations with it, and also get people to understand how this fish life starts. Because a grouper also starts as an egg that's just 0.5 millimeter big. And in the end, it can be this gigantic fish, but if you don't know how they look like in the first days, you just don't know anything, in my perspective. And what we hope to achieve is actually, in the end, make an autonomous breeding system. So we can make sure that it can get to the most remote places and you can leave it there and actually breed fish in open waters. But I also think we need a different approach in working together with local fisheries, because these guys know something that we all don't. I've learned more from the fishermen than any other organization. It's about fish in Bonaire. which is funny. And next to the fishermen, most of the things I learned was from Stinapa, because we're doing something in practice. And I think there is a good way of working that we also try to figure out for ourselves. And I call this the 3-pillar approach. So science is only 1/3 of what we do, because we try to make a nature-based solution, and it's more of a pragmatic solution, as Francesca also stated, than something else. Science is the other third, and engaging with local-based communities is the other one. And basically, the first weeks of me working in Bonaire was only doing the local-based community stuff. And it can even be small things. I remember running to Francesca because our pipets were gone, because we were in such a hurry. And then she helped me out. But this pipet makes sure that we can do this DNA analysis. It's only 50 cents, but to me it's priceless. And I think that also shows where we can get to. And especially with the fishermen, it really helps to work together with them. It really helps that you're also on the boat together with them. Probably you will puke, I did too. But it gave me such a good understanding of what they are doing and how we can use what they know for our advantage. But also on the other hand, for example, we don't know too much about the East Coast, because not too much people want to go there with the boat. So if Erik Meesters, for example, wants to do research, he needs to go to the fishermen and go to the east. Why don't we use this? These guys dare to do some things that we don't, which makes them valuable, but they don't know it yet. And I think that's very important. First, come up with the practical solutions, then make sure that we can make science with it, and not the other way around, because I think that's why we don't value sometimes even the national parks or the fishermen that much, because the most time I asked from people, I also occupy space with DCNA. I also occupy some time management from the WWF, because we want to fit into the framework, and I'm really glad that we are now in the strategy for the coming 5 years together with WWF. But if it's about time, it's the rangers, it's the fishermen. That was the most time-consuming thing for us, but also for them to help us. And I think that was where the power laid in this project, because we know where we should be, we know how we should get there, and we also know what's still needed. And if we talk, for example, about the science-based part, a lot of that will just be able to make sure that we know more about abiotic factors. And I think it's just as much important as it is to me as it is for example, for Francesca, because if we know more about surface currents, we actually know what happens with these eggs, but most measurements happen underneath a buoy, so you don't know anything. So let's try to work together and make sure that at least, and I think DCNA can really have a role in that, and or Stinapa, to make sure that we have an overview of what's happening on the island and make a comprehensive approach together. Because if I have an aquarium and I would say, Francesca, you do the corals in my aquarium, Alwyn, you do the sea urchins, you can do the mangroves, Arno, and you don't talk to each other, it's getting worse than if we all work together. And I really hope that we can achieve that. Here, I have a still. Sorry for the people at home, I won't show it now, but afterwards I will show it to all of you, because I was talking about making movies. This is a parrotfish coming out of the egg. And the coolest thing is, if you look this up at YouTube, you can find it. And I think there lays the power of the story also with reef renewal. It's what Jamie does. If you see how these early life stages start so small, and then in the end it builds these metropoles of reefs that we all like so much, we get a better, broader understanding of how fragile their life starts. And I think that's what's needed in the public. The fact that I could show fishermen the first day of the fish that they were actually catching really broadened their experience. But they were one of the first to actually see those, and there lays also the start of a relationship that's different for them, because they're just as much as a partner as DCNA, Stinapa, WWF. It doesn't matter. If they don't contribute in money, but in knowledge, it's still the same. And I'm so glad that I can do this, but also I'm so glad that I can show you, I hope, a different way of working. And not in everything, but in some things it is, I hope. I really want to thank WWF, because without their involvement, we would never be in Bonaire, and maybe I was still stuck in the aquarium with the wildest dreams. DCNA, because I can give this presentation here now, but also I can walk into their office and do my weird, funny fish thingies. Stinapa, because all of the times we ran out with the boat and made sure that we could catch fish eggs in different places where you normally can't come, because as you may know, we swam to all these places, so it was a good workout all of the time. And Piscabon, because all of their knowledge hasn't been written down before, which doesn't make it science, but it can be science in the future. If you want to know more about the Rotterdam Zoo plans, because we have a whole different direction and goals, you can scan this QR code. Basically, us as a zoo, we think we are not a goal, but we are the means to achieve the goal, and that's, for example, here in Bonaire, healthier reefs. Thank you for listening. Are there any questions?
Thank you so much. Yeah, opening the questions first to people.
Thank you very much for the presentation. I was wondering about the eggs, because you see the differences between the different species, but how sure are you that within the same species, all the eggs are the same?
Well, that's basically what we're finding out still. So I will show it in the afternoon a little bit better. Sorry for the people at home, but we don't have all the time. I can talk hours about this, but what you see is mostly eggs that are between 16 and 24 hours old. So if we really want to have a comprehensive approach, we should track this all the time. And that's why I want to make movies, because fish eggs are really fragile, so they die quite easily. So now it's making the picture, release them back into the water, and you don't want to mess around with that too much. But if you want to measure growth and the first stages, it's really important that we get to the next phase there. But for your information now, for example, we have 2 students here running the work. It's not enough to also have that approach if you want to increase the numbers. So in September we will have 5 interns, and then we can do more testing of the in-between phases. That's why I also just want to focus on the first, only the eggs and the hatch, and then afterwards the hatchlings, because otherwise we really get lost in, I always call it Candy Mountain, because it's super cool to try to tackle it all, but you also have to have a pragmatic approach if you see these things for the first time, of course.
Do you catch other things except fish eggs, like
yes.
coral eggs? Or...
Yeah, we catch a lot of coral eggs, and that's why I think it can be cool if we optimize the system, and that's what I'm trying to do right now. So, like I said, I'm more of making tools, and it's up to others to make science with it. But I think if we can make sure that we have a camera that can register it all day long, which is my goal for now, then we can really correspond the data. And I think I can reach out, for example, to Seek or to Francesca to make sure that we actually get a comprehensive approach of what is happening in the sea. And for example, sea urchins, we don't see that much, but it's also because of the way we look. So a good example is there's some fish eggs that look like honingraat, honey, like the bee ray. Yeah, honeycomb, sorry. So they look like honeycombs, but before we were only focusing on the fish eggs as we knew them. And then we found out that we were throwing away some fish eggs because it had a honeycomb shape. So it looked like a honeycomb all over, and we thought it's a diatom or something else. So that's also how far we are sometimes. Sometimes we don't even know what we're looking at. But if you look at everything, and I will tell this to the interns, then basically it's actually kids getting stuff out the lake. So we need to have some structure.
How many eggs do you need to collect for the PCR to work for the
one? So what we actually do is we put them in 99% alcohol and then test them in the lab. And now in the Rotterdam Zoo, we're trying to put up this lab ourselves. So the goal is that if others also want to take pictures and catch them, that we in the end, so we're not near there yet, but we are open for some testing together with others, see if they can make a picture, then send a DNA analysis and see if we can build up this database. That will be the ultimate goal, because I think then we get a real understanding of how these fish spawning actually works. For example, if we put out the nets, I think you sometimes see the same with seek or reef renewal. If you have something floating around the water, it's always going round. And we always assume some things about fish which I think are not entirely true. For example, if a fish is born here, some of them go to Curacao, but I think the main portion of them stays on the reef. But we don't have too much abiotic measurements to actually prove that. Another thing is mangroves, because I think there's a huge misunderstanding about mangroves when we call them nurseries. There's only one scientific paper that I could find till now where we can see that a fish actually breeds in the mangroves. So it's basically or kindergarten or the bike rack behind high school, but I don't see a difference there. And that's basically because we don't know anything about the first life stages of fish. So unless we have proof, we should also try to think outside of the box, or outside of the box fish, as I say sometimes. We really don't know how it works yet. So first start with that and then draw conclusions. And now sometimes we learned ourselves conclusions that is based on nothing except we see small fish. But a small fish is only what we can capture with the eye, as you can saw. And probably we'll see this afternoon, you can't catch many things with the eye in the first early life stages in the ocean. So that's really hard. And it's still also hard for me, because now it's harder for me to look them up again. So probably the interns are better at looking at fish eggs than me right now.
You joked about the gardening of eating a tuna. I actually think you should really look into that, because I listened to a talk about 10 years ago by Osaka Aquarium that bred their tuna. And the way they did it is they reared one cohort and then they cooled down the rest and they fed the ones that they slowed down to the others, and that's how they go to it.
Yeah. Well, that of course that happens a lot in. I think some people even do it with anemone fish if they are running out of artemias, because after a while you need to grow them with you. So anemone fish are quite easy to breed. So sometimes when some people are short on supply, they just wait and then feed the anemone fish to each other. So it's quite common for fish larvae to eat other fish larvae. We already know that. But like I said, we just don't know what their preferences is, and that's the main hurdle we need to tackle right now in aquaculture on land. And I'm convinced that this will make us learn way faster. And what the outcome is, if we actually will restock reefs with fish too, it's my dream. And I think you only know if it works if you try it, and not the other way around.
Yeah, one more, and then we have one from the Zoom participants.
Well, thank you for the inspiring presentation. I still love your passion for fish.
I think
it's quite niche, so I love it. I want to ask you, so I think you open our eyes to a lot of different application possibility, the vision for what we do, and what you're doing is a huge, really endeavor thing. Once you complete this phase, which is, again, I'm not underestimating, like the ID phase of all these eggs, what do you think would be, maybe you said it and I missed it, what would be your ideal next step?
Well, I think first getting numbers, but as I said, because it's also about an endeavor, you don't know. I had a lot of talks, for example, with WWF. How can we make a key KPI about this? But actually, we don't know if it will go as fast as this in the future, because it can be that we have to differentiate in, for example, depth. So we know that these oil globules are there to make the eggs float, but now we only take the surface, and I think we also probably we need to have a free dive line and then go all the way down. So there's, like I said, there's the two roads that we actually are going for right now. So just proof that a system works where you actually move along with the sea. I think we already have some proof, but I need to make that scientific. Sorry, I forgot to say, that's not my role, of course. That's what I already had the pragmatic proof, and now it's up to science to do the rest. But I think there lays also our first hurdles where we actually want to measure more. And that's why I'm talking about this microscope. I really need to make sure that we can track what is happening in the sea in these early life stages. And then we can also tell more about hatchling phases, about breeding them further. A good example, for example, is these eggs in gelatinous ribbons. Because that's a way that I think, in percentages, I'm not sure, but a lot of other fish are also breeding, and they still have these oil globules. So probably it's not up to 80%, but even more percentages of fish that actually start their life floating, and basically it's not a real larvae yet. So understanding how that works and why it works like that would really help us understand to take the next steps. But it's very hard for me to oversee, where sometimes I'm also lost in all opportunities. So that's why I'm focusing on eggs and hatchlings first, and do some trials on the side. And that's why I also try to make these 3 pillars. So basically, I'm not into science, but because I make the two, I will still help with reporting, and you understand how it works. But I think there's also, and Jamie stated it too, there's the power of aquaria, where we can think of a practical first step. And I think it's the same for you guys. I think, and then I finished. I think the best explanation of aquaria is we kickstart a lot of things. Dirk Pedersen did his PhD in the Rotterdam Zoo, and when I talked to him, he saw a lot of similarities in what he did and what I did. But basically, it's not to make it cooler or better. It's C-Core 2.0 in what you need to build, because the coral is not moving, the fish is moving. So it's more different steps where you first need to build more things. And basically that's why we have this strong partnership with the Technical University Delft. I'm more of an inventor, I always say. And that's also something that we need to apply better. So we need to make sure, for example, that WWF knows that we have a fail rate. We need to have like a fail rate of 80%, because otherwise you don't know what works best. And I think it's the same for you guys, but you already proved your methods a bit better than I did, of course, because I'm only starting for 2 years.
Thank you so much, Sander. For the people at Zoom, we're going to get the question.